Today I'm going to present my research on HPTLC, a good choice for extractable and leachable study. So let's see. So this is the brief outline of my presentations. So basically my presentations divided into three parts. First part is extractable and leachable study for phthalates for pharmaceutical product. Second part is biomonitoring of these leachables uh, uh, is a metabolites, and third one is a comparison of in-house method developed for HPTLC and LCMS. So let's start with the first part. So basically, it is believed that drug container and modern drug delivery system, it's meant to protect the drug from the environment, and uh, and it prevents to contaminate products from the environment, but actually themselves as a source of contamination many times. As we can see that the many pharmaceutical preparations are packed in plastic container, and when it packs with plastic container, the plasticizers that are used to make a plastic that are freely bound to the container, and this container, uh, this freely bound component can be easily leached into the product, and then it will contaminate the product, and sometimes they are harmful. So what is extractable and leachables? So extractable means test the material, leachable means test the product. For any organic or inorganic chemical entities that can be released from the packaging into the product, it is called as a extractable and leachable. So why we have to worry about extractable and leachables? It can affect the efficacy, safety, and quality of the pharmaceutical product, and it is also I have a data like in 2014 FDA has recalled the drug. 836 product recall has been made due to the safety and quality issue. And in European Union also, it is the third most significant category for product recall. That is approximately 14% of the total that are corresponding to the problem with the packaging material. So it is quite a serious concern as per my perspective. So let's see the what are the harmful compound identified as extractable leachables till now. So one category is phthalates, toxic elements, polyaromatic hydrocarbons, nitrosamines, and out of phthalates, uh, there are phthalates is a category. There is a diesters, and in diesters, the compound very popular compound is dimethyl phthalate, diethyl phthalate, dibutyl phthalate, and so on. So these all are used as a plasticizer to make a plastic flexible, durable, and consistent. So what are the side effects from these plasticizers, or we can say phthalates, or we can say extractable and leachables? So it has a side effects on endocrine disruptor. It may cause male infertility and decrease sperm count. It has anti-androgenic properties. It irritates lungs. It has a genotoxic effects too. And out of these all phthalates, DBP and DEHP like diethylhexyl phthalate are the most toxic compound. So what are the regulatory guidelines for pharmaceutical packaging? So several guidelines has been made by the US Pharmacopoeia, by European Pharmacopoeia, and ICH. They all are providing the guidelines that how to use the packaging, and if you use the packaging as a plastics, then what are the risk assessment you have to do in terms of the extractable and leachable study. So these are the regulatory guidelines which will control the plastics in product. So as we done the literature survey, there are many popular techniques for this kind of study that are GCMS and LCMS, MS for toys, food, consumer, cosmetics, medical device, pharmaceutical, and waters. But as far as HPTLC, there is very less efforts has been made to prove that these methods are this method can be useful for the this kind of study, extractable leachable. So we did the first efforts. We did the efforts for this extractable leachable study. We have selected the four phthalates, that is dimethyl, diethyl, dibutyl, and diethylhexyl phthalates for our study, and we have succeeded to separate all these four phthalates as a from the as a standard. And for the sample selections, we have procured the samples from the local market, and we have selected the product. Many of our products are the parental preparations because the leaching probability is more in case of pharmaceutical preparation. So we have almost selected all the selected preparations are parental preparations. So that has been selected, and if we in our study, and the sample ID is given as sample 1 to 13. So sample analysis protocol, what we have did for the sample analysis. So this is sample preparation part. 30 ml of product has been taken, and it was extracted with 20 ml of annexane after in separating funnel. After separations 2 to 3 times, organic layer was collected, and this procedure has been repeated for 2 times. And this organic layer was evaporated to dryness at room temperature, and it is reconstituted to 1 ml with methanol. And as far as extractable, we have to extract the container. So we have take the 2 gram of plastic container and cut it into smaller piece and extracted with the 30 ml of distilled water and sonic it, and rest of the procedures remain same. So in first part, we have tested the product. Second part, we have tested the container. So as far as result is concerned, so all four phthalates has been separated very well, as we can see in UV photograph of TLC, as well as it is in densitogram of at 240 nanogram. All compounds are well separated, and later on the all samples which has been selected for our study, that has been also applied. So you can see the significant amount of phthalate content, selective phthalate content has been there. So this is a method validation summary. That is a linearity range, equations, precisions, recovery, LOQ. So we have succeeded to do the analysis from 100 nanogram per band, which is quite sufficient for this kind of study. And looking to the data of precision and accuracy, we can say that the method can be useful for the desired applications. After the sample analysis, what results we are getting? That is for DMP dimethyl phthalate, the 5 sample has been found as the leaching content, extractable content, and within the range of given. In the DEP diethyl phthalate, we have found one component as a extractables, or 2 samples found in the range of in the category of leachables. For DBP, dibutyl phthalates, again, we have found 5 samples content in extractables, and 2 samples the DBP was found as the leachables. In DEHP, almost you will surprise, I mean, we will surprise that all the samples are having a content of DEHP, which is most dangerous, and we can say carcinogenic compounds amongst the all phthalate category, and which we have found in all the products. So as per concern of PQRI toxicology, team proposed that there is a has classified the class 1 to class 5, and what is the threshold level that is required for per day in microgram per ml, microgram per day. So DBP means dibutyl phthalate and diethyl exyl phthalate were dominantly present into the product, and which was considered as a more toxic as a class 1 compound, and whereas DMP and DEP were also found, but it is within the threshold limit. Okay? So now we have results that the plastic is found in the uh, pharmaceutical product, but we wonder that whether when it reaches to the body, what happens to the phthalates. So in another kind of research, we have extended our research study that the plastic is now being very common in our day start to day end. So we cannot avoid the plastics from our life. Okay, there are many route of ingestion of plastics, from the inhalation, ingestions, and dermals, the milk, the water, the fast foods, everything is packed in plastic. So uh, when it gets to the body, we have to identify the plastics from our body. So whatever the phthalates that are going to uh, in the body, or that will metabolize in the body and in the liver, and the more than 95% of the metabolites are excreted from the urine in the form of monoester metabolites. So it is dibutyl phthalate, so it is converted into the monobutyl phthalate. So diesters becomes a monoesters, and it is excreted from the urine. So we, in further study, we have selected the monoesters as a metabolites for the routine monitoring. So this was DMP was converted into the monomethyl phthalate, dibutyl is converted into monobutyl, dibenzyl converted into monobenzyl, and DEHP converted into the monoethylhexyl phthalates. So that is, in second part we have selected these four metabolites. Okay, and we have prepared the samples according to like these, 1 milligram per ml, and the enzyme is also used for deconjugations. These are the sample preparations. We have used the ethyl acetate. Uh, we have used the enzyme. We have used the ethyl acetate as a extracting solvent, and we have also checked what is the effect of enzyme. And so, in first study, uh, sorry. So this is the data of deconjugation of metabolite with the use of enzyme and without the use of enzyme. So you can see the recovery. The recovery is very less when we have not used the enzyme. And when we have used the enzyme like beta glucuronidase, so it is literally deconjugate the metabolites from the urine matrix, and it will increase the recovery. And it is almost more than 40% of recovery in each case we have observed. So that's why we have used the deconjugations enzyme, that is a beta glucuronidase enzyme. Then for ethyl acetate, we have selected the different organic solvent for this study, like dichloromethane, TBME, diethyl ether, ethyl acetate, and hexane. And uh, at the end, we have observed the significant recovery in terms of ethyl acetate than the other extracting solvent. So this is the real experimental work. We have separated all the phthalates, four phthalates, and the good chromatographic separations was achieved, and it is also presented over here. We have also received the good linearity in terms of phthalate content in a urine matrix, and these are the validation summary. Uh, that is all the linearity was more than 99.99, so it is a good conditions. Then looking to the precision accuracy data and matrix effect and stability, it can be further utilized for the bioanalytical methods for routine monitoring. Then we have not restricted our study to only the method development. We have also do the reverse analysis, because we have already found that from the pharmaceutical product, the phthalate content was already there. And the major side effect of this phthalate was making infertility in the human. So we have made up one hypothesis that we have to take a samples from the ICU patients, and we have also selected the patient uh, uh, who are undergoing the in vitro fertility treatment. So in ICU patient, it is ensured that the parental preparations are ongoing, and in IVF treatment, that is the side effect of the phthalate. So we have totally selected 51 samples as a populations, and these are the sample analysis results. So we have seen the in many patients we have selected this phthalate metabolite content in their urine. So it is a summary of sample analysis, like in ICU, out of 14 samples, 5 samples has been discovered with a monomethyl, monoben, 2 is monobenzyl, and 2 was monobutyl. And as I said, DEHP is a dominant plastic, dominantly used in the plastic making. So it is also uh, uh, receiving in a large number of patients, like 10. Okay, and percentage of patient exposed to the phthalate content means almost 71% patients are getting detected with the phthalate metabolite is MEHP in ICU. In in IVF treatment, out of 37 patients, percentage of patient exposure to phthalate metabolites, almost 60% of the exposure has been made in terms of monobenzyl and monoethylhexyl phthalate. So we can say that there is a significant amount of phthalate content was found into the sample patient. This is a sample analysis content, the lowest value and highest value. So you can see microgram per ml in the urine and highest value. And we have also further classified that so many patients are in the uh, very high amount of phthalate metabolite in their urine when we analyze. So, so it is it makes lots of things in our mind that how uh, the HPTLC has used to discover these all those things. But previously, when we started HPTLC, before that, we have also used the popular technique in 2015 with the LCMS-MS. So this is in-house developed LCMS-MS method for extractable leachables. So we have, by doing the literature review, we have found that this LCMS-MS is the most popular techniques for this kind of study. So we have also did this, but after doing this study, we found that because plastics is now everywhere, the solvent which we are using, the water which we are using for the analysis, everyone is sometimes get exposure from the plastics. So when we use the most sensitive technique, at that time we are getting the mass saturations or baseline from the system. So that's why we are getting the baseline up when we are going analysis. So at that time, sometime we are losing the sensitivity. So what common practical hurdles with LCMS when we have analyzed? So due to baseline saturations, it might be from our part, but we have excluded DEHP because of the mass saturations and which is more prominently present into the product. Okay, but still we have to exclude because if we include that then we have lose the sensitivity. So by doing the comparison of HPTLC and LCMS-MS, what we have found that sensitivity in terms of sensitivity, the HPTLC is enough sensitive like 100 nanogram per band. In LCMS-MS it is more sensitive. It is of course LCMS-MS is known for this. Number of phthalate analyzed, we have analyzed the 4 phthalate in our study, while in case of LCMS-MS, we have restricted to 3 because of the we do not want to sacrifice our sensitivity. Method development, it is a simple. In LCMS-MS, it is quite uh, complex. Method validations, in both case, it is complied with the validation parameters. Sample analysis, yes, we can analyze, we can quantify in the both case. Sample processing, yes, it is also simple process. Instrumentations and cost wise, also HPTLC have its own benefits. So we came to conclude that in LCMS-MS, there is a possibility of mass saturations, complex sample preparations, more number of optimization parameter, complex instrumentations, and greater cost for the such type of study. Uh, when we compared this technique with the application and cost, so it will also come to know that LCUV and these all the techniques have their limitations. Like in LCUV, chromophoric compound is required, GCMS volatile compound, LCMS non-volatile compound, and ICMS is elemental. And with respect to cost, also it is HLCMS having approximately 80 time cost than the HPTLC. So I can conclude that the HPTLC is more fit for purpose because it has a now option for TLCMS also. So we can further confirm with the mass and we can conclude to the for the study. So as a conclusion, HPTLC is an essential instrument and fit for purpose solution meeting the regulatory aims without over engineering for extractable and leachable study. This technique can be also useful for the routine quality control tool which significantly minimize the cost. And a sufficient analytical evaluation threshold has been also established with suitable for safety threshold for phthalates with HPTLC techniques. And HPTLC can effectively utilize for biomonitoring of phthalate metabolites from the human. So in nutshell, I can say that HPLC is also fit for purpose. So as the first part, we have applied for the patent in the Indian government for the HPTLC method for the detection of phthalates, extractable and leachables from pharmaceutical product, and we got the patent for this study. And another part, the HPTLC method for detection of phthalate metabolite in biological material, it is still under evaluations. It is already published, but it is under evaluation. So we are also hopefully for these methods to be granted for patent. Thank you very much.